Good day, everyone. We're reviewing Wetzel's Limnology textbook. This is edition number 4, and we are on chapter 9, Structure and Productivity of Aquatic Ecosystems. And more specifically today, we will be looking at subchapter 4 that deals with spatial structure and terminology in lakes and rivers. So let's get started. In rivers, a range of different terms are used. The riparian zone refers to the riverbanks and their vegetation, which can tolerate periods of inundation during floods. Rivers may be divided into headwaters, small erosive streams at the upland end of a catchment, middle reaches, and lowland rivers. However, a variety of different geological forms may exist at different points along rivers. Therefore, rivers are often divided into a nested hierarchy of structural forms. These include river segments divided by tributary junctions. Nested within these are reaches divided by structures such as waterfalls, debris dams, or large pools. Nested within reaches are habitats such as pools, runs, and riffles, each of which is defined by relative depth and flow speed. Pools have very slow flow and are deeper than runs, which have faster, smoother linear flow, and are also deeper than riffles, which are shallow with fast turbulent flow created by stones. So let's look at these two figures here. Figure 9-3. So we're looking at the terms pertaining to the land lake interface. So the profondo term is used for the area at depth. And then we have the littori profondo, which is above the profundal zone. So it's pretty much like the littoral area of the depth of the area at depth. Then we have the littoral zone, and then the supralittoral zone, which, as you can see, is above the lake water. And then on the right, we have the pelagial zone, which refers to the aquatic environment, the terminology used in the aquatic environment. And here we are looking at a watershed system. So this is the ridgeline. And so any water falling on this side of the ridgeline will end up either in groundwater or used by biota or evaporated, or some of it will be reaching the rivers themselves and the streams. And then they are magnifying this area. So we have a ridge system, and the ridge system in the order of 10 meters, and then the riffle pool system in the order of 1 meter. Some terms are used in both rivers and lakes. The term benthos, originating from the Greek word for bottom, is defined broadly to include the assemblage of organisms associated with the bottom, or better, any solid liquid interface in aquatic systems. Periphyton, although variously used, usually refers to microbial and or algal growth upon substrata. It is a broad term that applies to microbiota living on any substratum, living or dead, plant, animal, or non-living. Biofilm is another term more frequently used to describe much of the same thing. It refers to attached communities of algae, fungi, and bacteria growing on any submerged surface. Subchapter 6. Subsidies and productivity in lakes and rivers. The living biota of inland waters constitute only a very small portion of the total organic matter of ecosystem, even in the most productive of freshwater habitats. Most of the organic matter in aquatic ecosystem is non-living and is collectively referred to as detritus. Detritus consists of all non-living organic matter in both dissolved and particulate forms. In aquatic ecosystem in general, nearly all of the organic matter consists of dissolved organic carbon, DOC, compounds, and particulate organic compounds, POC. The ratio of DOC to POC is usually between 6:1 and 10:1 in both lake and running water ecosystems. The functioning of aquatic ecosystems centers on the cycling of organic carbon between living and non-living components. Because of the pivotal relationship between the productivity of the living components and the massive amount of non-living organic matter, the subject will be discussed in detail in these chapters. Autochthonous productivity means carbon fixed within a water body by microbes, algae, and aquatic plants. Allochthonous productivity arrives in the water body through terrestrial subsidy. In most rivers and lakes, both forms of subsidy occur, with the relative importance depending on the morphology of the water body, the geology of the catchment, and the abundance of riparian or fringing vegetations. Autochthonous production may be carried out by microbes, photosynthetic bacteria, algae, or aquatic plants. These photosynthetic organisms all require light to fix carbon. Therefore, freshwaters that have low light availability through shading by vegetation or through the presence of dissolved organic matter or inorganic particles, decreasing water transparency, will have little subaqueous autochthonous production. However, this does not mean that autochthonous production is not important in those water bodies. In turbid, intermittent rivers during the dry season, a thin ring of algae and cyanobacteria grows in the shallow water at the edge of perennial pools. Although teeny, this highly productive bathtub ring supports the entire ecosystem. Where there is sufficient light, the form of the substratum and flow speed determine which photosynthetic organisms occur. For example, many aquatic plants have roots in the substratum and cannot grow on bedrock or among boulders. If they also have foliose leaves, having numerous broad leaves, they will not be able to persist in fast currents. For these reasons, autochthonous production in stony, fast-flowing rivers is done by microbes and algae that live in the biofilm on stones and bryophytes. Low-growing tough plants that can attach to stone and withstand fast flows. In lentic water bodies, such as lakes, no flow, or slow-moving rivers, mid-lowland rivers, substrata are often finer and more suitable for the roots of aquatic, including foliose plants. If there is sufficient light, these stands of microalgae and vascular plants will grow. Depth is also a factor here because some aquatic plants require shallow water to obtain sufficient light for growth. In deeper water, most autochthonous production will be done by phytoplankton. These are algae and microbes capable of floating or swimming in the water column to obtain sufficient light, but can be washed out during fast flows. So figure 9-5 illustrates that bathtub ring, and a thin ring of algae and cyanobacteria growing in the shallow water at the edge of perennial pools. So right here, I think this is what they are referring to. This highly productive bathtub ring may support an entire ecosystem. Allochthonous productivity arises in the water body through terrestrial subsidy from terrestrial or fringing plants. Allochthonous organic material is divided into size classes: dissolved organic carbon, less than 0.045 millimeter, fine particulate organic matter, FPOM of 0.045 to 1 millimeter, coarse particulate matter, and then large organic matter. FPOM and CPOM enter rivers and lakes through leaves, barks, or nuts falling into water bodies. LPOM enters water bodies through branches or entire trees falling in. DOC is released by the leaching of all forms of organic matter when they enter the water. DOC may also enter a water body when soil that is rich in organic matter becomes inundated and organic matter dissolves, leaches out of it. By darkening the color of water, DOC can limit photosynthesis and therefore autochthonous production. Furthermore, it is itself a food source for aquatic microbes and in high concentration can stimulate heterotrophic carbon pathways in aquatic food webs. F, for fine particulate organic matter, is consumed by a wide diversity of freshwater invertebrates. It may be nutritionally enriched by presence of microbes on it, and it also includes fecal pellets from invertebrates and pollen from riparian plants, which is very nutritious apparently. CPOM is colonized by a biofilm so CPOM is coarse particulate matter, is colonized by a biofilm of microbes, fungi, and algae, which increase its nutritional value to the few invertebrates shredders who can consume it. CPOM also provides a habitat for invertebrates, whereas LPOM, or woody debris, also provides a habitat for larger animals, for example fish, in addition to invertebrates. Rivers vary in the predominance of autochthonous or alloctonous production. The first model to recognize this was the river continuum concept, which predicted that as headwater streams were erosive and shaded by forest, their ecosystem depended on alloctonous carbon. Further downstream, rivers widened, so they was assumed to be more light, thus increasing autochthonous production. In their lower reaches, rivers are generally much wider and deeper, and so phytoplanktons were thought to be the dominant producers, but considerable amount of fine particulate organic matter were also deposited in those reaches from sources upstream. Later research has shown that there is considerable variation in the balance between autochthonous and alloctonous production along and between rivers, and that algal carbon is usually the best quality carbon source for consumers even in upland streams. In lakes, alloctonous subsidy may be less important than in rivers due to their larger surface area to perimeter ratio. They still receive CPOM, coarse particulate matter, or LPOM, large particulate matter, but these sources of carbon are considered less influential than in rivers. The emergence of fatty acids and stable isotope research shows, however, that terrestrial particulate organic matter can enrich phytoplankton-based food webs, with the importance of terrestrial subsidy increasing with decreasing surface area to perimeter ratio in lakes. Evaluation of biomass and production. In order to evaluate the productivity of a system, it is important to realize that production is a rate and is often expressed as a unit of mass or energy production per volume, per time. Confusingly, biomass or standing stock is often used as a proxy for the productivity of a given system, but this is inherently wrong. Biomass is the mass or weight of living material in a unit area at a given time, instant in time, and is often calculated as the product of mean individual weight, W, and density, N. Biomass has the dimension of mass per unit area or volume. Biomass can be calculated for certain functional groups such as algal biomass, zooplankton biomass, macrophyte biomass, and fish biomass, and as such be used for calculating the productivity of these groups. Another useful term in the context of productivity is carrying capacity, the maximum biomass a specific group or even an ecosystem can achieve given the prevailing abiotic and biotic factors. Similar to biomass, production can be evaluated for a single species or a specific group, but also on higher organizational level up to the ecosystem level. Primary productivity. The primary production of phytoplankton has received an extraordinary amount of attention in limnology and has been measured in great detail in a number of aquatic ecosystems. In a process called photosynthesis, autotrophic species such as phytoplankton produce biomass by using light as a source of energy to convert CO2 and H2O into glucose and oxygen. Respiration, on the other hand, is a process where glucose and oxygen are used to produce CO2 and energy, whereas gross primary production is defined as the total rate at which autotrophs produce biomass or energy. Net primary production is defined as the rate at which autotrophs produce biomass or energy considering the energy or biomass used for respiration. The light and dark bottle technique for estimating primary production have received broad applications. In the oxygen method, samples of phytoplankton populations are incubated at the depth from which they were collected in clear and opaque bottles. The initial concentration of dissolved oxygen, C1, can be expected to decline to a lower value, C2, by respiration in the darkened bottle and increase to a higher concentration, C3, in the clear bottles, according to the difference between photosynthetic production and respiratory consumption. The difference C1 minus C2 represents the respiratory activity per unit volume over the time interval of incubation. The difference of C3 minus C1 is equal to the net photosynthetic activity, and the sum C3 minus C1 plus C1 minus C2 is equal to C3 minus C2 corresponds to the gross photosynthetic activity. So let's look at figure 9.6 here. Light and dark bottle technique for estimating primary production. So the bottle wrapped in tin foils are the dark bottles right here, excluding light. So there's no light coming into these bottles, and allowing the assessment of respiration. So there's like pretty much like a water sample in there that was taken at that depth with the material that would be found there. And then these are like the bottles that are clear, and there's no foil, there's no metallic foil on it, so the light will be reaching the biota in here, and then here, therefore, you're going to have, instead of respiration that you have in this bottle that are darkened, here you have photosynthetic activity, so you have production of oxygen, for example, here. Numerous assumptions are made in the method that can alter the photosynthetic measurements appreciably. For example, respiration rates are not necessarily the same in light and dark. Since photorespiration clearly occurs in algae, other processes such as photooxidative consumption use oxygen separately from apparent respiratory uptake, non-photolysis of bacteria by bacterial photosynthesis, and so forth. Under many circumstances, these errors are small, but the technique can be considered only as a reasonable estimate. It is probable, however, that these and analytical errors in the determination of oxygen concentration are appreciably less than sampling errors in the analysis of heterogeneous plankton population in the lake. Large portion of methodological works are devoted to detailed discussion of this and the following C14 techniques. The incorporation of C14 tracers into the organic matter of phytoplankton during photosynthesis has been used as a sensitive measure of the rate of primary production. If the total CO2 content of the experimental water is known, which can be estimated from a lake water alkalinity analysis, and if known amount of carbon-14 CO2 is added to the water, then the determination of the content of labeled carbon in the phytoplankton after incubation permits the estimation of the total amount of carbon assimilated. Numerous methodological and physiological problems confront the application of C14 light and dark technique. With care, however, most technical problems can be overcome and errors evaluated. For example, respiratory losses of CO2 and secretion rates of soluble organic products of photosynthesis. Rates of respiration are difficult to evaluate directly with this technique. In many situations, the C14 method yields a measure close to the net photosynthetic rates. Comparison of the oxygen and C14 method under optimal conditions shows close agreement with the photosynthetic quotient. For example, the volume of oxygen released in photosynthesis as a proportion of the volume of CO2 used in that process. The photosynthetic quotient varies from near unity when carbohydrates are the principal photosynthetic product to as high as 3.0 during the synthesis of lipids. Assuming a photosynthetic quotient of 1.2 and a statistical significance level of P less than 0.05, the smallest amount of photosynthesis that the oxygen, light, and dark techniques can measure under ideal condition is about 10 milligrams per centimeter cube per hour. The limit of sensitivity of the carbon-14 method is some 3 degrees of magnitude lower, on the order of 0.01 milligrams per centimeter cube per hour. Secondary production. Secondary production in fresh waters by invertebrates and vertebrates is much more difficult to estimate accurately than is primary production. Trophic relationships are complex and often change during the life cycle of a species or from ecosystem to another. The size of animals varies greatly from immature to adult stages, and the diet can also vary greatly throughout their life history. Because most animals are mobile, they actively distribute themselves in response to environmental stimuli, which results in heterogeneous distributions and make accurate sampling more difficult. Production measurements of animals are based on the estimation of numbers, biomass, and growth rates. The sampling accuracies of changes in population size decrease from zooplankton to benthic organisms to fish. In contrast, growth rates are easier to evaluate in many temperate fishes and in long-lived invertebrates than they are in small, rapidly reproducing zooplankton, other invertebrates, and protists. When evaluating the population dynamics and production of any animal, estimating food incorporation or ingestion and the utilization of ingested food is imperative. Assimilation means the absorption of food from the digestive systems, and the efficiency of assimilation refers to the percentage fraction of ingested food that is digested and absorbed into the body. Assimilation efficiency is not constant and varies greatly with the food quality and rates of food ingestion. Measurements of assimilation are based on the simple relation of assimilation is equal to ingestions minus exgestions, or assimilation is equal to growth plus respiration. Although these relationships are simple, their accurate measurements among natural populations of zooplankton and larger organisms is problematic. So the method employed I discussed quickly by these authors. Although ingestion can be measured in situ with reasonable sophistication, accurate in situ measurement of exgestion rates is complicated by inter-individual and temporal variation. Around the turn of the millennium, these conventional in situ measurements were replaced by stable isotope analysis used to estimate time-integrated trophic positions and biomass flows to consumers. Elements such as carbon, nitrogen, or oxygen can occur in different masses or isotopes in the environment due to the chemical and physical processes. To exemplify, carbon-12, carbon-13, and carbon-14 are three isotopes of the element carbon. Stable isotopes, as opposed to radioactive isotopes such as carbon-14, do not decay in other elements and are therefore used as tracers in food web studies. In aquatic systems, frequently used tracers are carbon-13 and nitrogen-15, which occurs in nominal amounts in the aquatic environment. These stable isotope tracers are compared to the more abundant and lighter stable isotopes carbon-12 and nitrogen-14 in stable ratios. For example, delta of nitrogen-15 and delta of carbon-13. As consumers typically become enriched relative to their food sources in delta N15 by 3 to 4 per mil. This stable nitrogen isotope ratio is often used as a time-integrated indicator of biomass flows in a food web. Stable isotope ratios of carbon-13, on the other hand, are more conserved throughout the food web, but do show distinct variation between the carbon energy sources of the food web base of primary producers, and are often used as an indicator of terrestrial aquatic subsidies of aquatic food webs. This is possible because aquatic plants, algae, macrophytes usually have different carbon-13 signatures than terrestrial plants. Using nitrogen-15 and carbon-13 as stable isotope tracers also allows for direct measurements of consumer assimilation and ingestion in experimental settings. So that will be it for today, and we'll pick it up tomorrow with ecosystem metabolism. Thanks for your attention, and talk to you tomorrow. Thank you.